Hi, welcome.
Hi there.
We're just waiting for Severine actually, so I'm going to give her a couple more minutes to sign on.
No rush.
It also works well, I know, for folks transitioning over from one of the other seminars, like for MCB.
That's right. Very good. Thanks for setting everything up.
Absolutely. Thank you for joining.
So there's still going to be some kind of discussion at the end?
Yep. Yeah, we'll have some Q&A and discussion amongst all the folks who are here. Yeah.
Excellent. Hi, Andrew.
Hello. I'm going to let you handle any discussion with Severine. I'm just popping in. So.
Okay, sure.
So
yeah, that'll be great. And I can moderate, and really we'll just have some Q&A from the folks who are attending.
I'm probably going to have to leave at some point. So.
No problem. I'm glad you could make it, especially with the back-to-back seminars today. I'm going to check in with Severine and make sure that she has the link and isn't having any technical issues.
So Andrew, your lab is functioning in shifts still with one-way streets down the...
There are no one-way streets. There are people in the lab. Basically, life goes on.
I gather the one-way streets they had in the like Stop and Shop and food places are largely ignored. Moment.
I've noticed that too, yeah. Although frankly, some of them feel very obviously posted, and then I notice some I have accidentally, when it's been a small arrow on the floor, I have accidentally violated it myself.
Yeah.
But I'm trying to keep a good lookout.
That's right. Some of them have hours for elders, and apparently that there can be real confusion when some of the over 70s are trying to negotiate the aisles. And Andrew, in your course, did you had lab in-person labs, or you mailed things to the students, or?
We can't have in-person labs, so the fall was computational biology, and the spring is microbiology, but it means that Emma's going to get them to read papers, and then she's going to show them experiments that she's going to get them to design experiments and execute some of them.
I see.
Well, let's see. This is when we'd normally be getting started. I've sent an email to Severine to hope that everything's okay at her end. And so I think what I'm going to do is at least show you the video that introduced me to this work in the first place, and hope that that gives a couple extra minutes of buffer time too, and then we'll see where to go from there.
Okay.
But I will share that from here. So obviously, David and Andrew, I'm sure you two are familiar with this video. It was a really neat thing for me to be introduced to a month or so ago.
It's a nice bridge between music and science.
Definitely, yeah.
Biology.
All right, so I think this is the best possible intro that I could give to this work. So I'm just going to let the video do the work. Here we go.
Okay.
All right. So that's Rocket East, which is how I first became familiar with this work. How, just as a little bit of background, how did you two start collaborating, and how did this project come about?
Well, I'll try and answer, but Andrew should interrupt, especially if I get the facts wrong. But I was fortunate to be involved in this quantitative biology an initiative that Andrew was largely running, and we've had, I guess, maybe a 10-year collaboration now, and fortunate enough to persuade Severine to come up the river from MIT and try to do something that bridged physics and biology in Andrew's lab, basically, with his very important help. And Andrew and I have been interested in these range expansions on solid agar substrates and how that plays out with spatial population genetics. And we decided to do it on a liquid. And Severine had just the right skill package and an interest in trying to do this bridging operation. And, but it was quite a challenge, at least from my perspective. I don't know if you want to add anything to that, Andrew.
Sure. So, I mean, this is a classical example of his life happens when you're thinking of other things. So David had done very nice theoretical work on how big flows in things like oceans could affect population genetics, but you can't exactly go out and do experiments in the ocean. And so it was his idea that you could maybe make an ocean inside a petri dish, but for it to be like an ocean, the flow has to be like crazy, crazy slow, because the flow has to be much slower than the time it takes cells to divide. And so he and Severine came and sat in my office, and David's like, well, you know, we can make the viscosity really high and pump this stuff and it will go really slowly. And I remember saying to both of them, like, I understand how all this will work in principle, but there's just no freaking way that you will ever be able to make flow that slow and make it that molassesy. And so then Severine came and made it that molassesy, but the experiments that David was originally envisaging, which required controlling the flow externally with pumps and stuff like that, and Severine did make devices to do that. We never got to those because the yeast made their own flow, and so we got distracted by something that we had never expected to see, and then had to spend time explaining it. And so Severine and a graduate student, she worked with Brian Weinstein, and they worked very well together, did this beautiful job of both doing experiments and figuring out what the heck was happening.
I think it took them more than 3 years to figure this out, and indeed the shock that Andrew's expressing that the yeast decided to make their own flows was all the more remarkable, because Andrew would know better, but I don't think they've ever had flagella in their evolutionary history, and so there was no obvious way that they could make the fluid flow, but it did. And they also managed to do some, with the help of Brian, some simulations that seemed to confirm the ideas about these metabolically induced flows.
Right. That was a really helpful explanation at the end too, of the video, that is, because I certainly found myself wondering how it was that without obvious routes of motility that was happening in the first place. Well, I don't know about you, but I feel like we have queued up the world's best teaser trailer for this talk.
Okay, so you see the message from Abby Plummer?
I did. I just saw that the internet has cut out. Oh no, that's terrible timing. That is one of the fears of doing everything virtually. and a very
Also,
real challenge.
working so there's I guess there's still hope. The other thing I would add, and
Yeah.
I'm interested in Andrew's perspective, is that one of the things we still need to understand more quantitatively is the influence of these flows on the population genetics. So when saw the kinds of movies that are in that musical introduction, and I thought of Polynesians going settling Micronesia with rafts, going across the surface of the real ocean. And, how does that affect their population genetics is an interesting question. And with these genetic markers from these lovely yeast strains that Andrew, who I regard as a kind of genetic alchemist, was able to get inserted, that's to me is still an object of fascination. And if you look at the colors in the movie of who begat whom, and what lineages, how the lineages interact with the sort of these tendrils sometimes that go out, or the fracture mechanics, almost like plate tectonics in the latter part of the movie. There are lots of open questions, and we're hoping to get other theorists interested, and maybe other labs involved in trying to
Definitely.
what happens.
I'm really glad to hear that background and that context for how this came about, because it's definitely something I've wondered about thinking about microbes in the ocean. It almost blows my mind to try to understand what constitutes a physical space in a moving fluid. So I'm glad that someone probably with much more background knowledge on that is thinking about it.
Well, I certainly don't have a lot, not an oceanographer, but, and there are big differences. As Andrew said, you have to make it like molasses so that the flow is slow enough in this tiny little environment so that the cells have time to divide and respond. There are some similarities. The ocean, most of the action, at least in my understanding, happens because of photosynthetic organisms, which, so they can get sunlight, have to be within 50 meters of the surface. So there's a relatively thin layer, at least on the scale of the ocean, where you have these microbial organisms like Senecacoccus and plankton, and absorbing light, doing their thing, and then being subject to oceanic advective currents, and turbulence. We certainly don't have turbulence in the Petri dish, but the eddy turnover times in the ocean are indeed long enough they can be anywhere from, say, 5 minutes to a couple months. The Gulf Stream is essentially perpetual eddy stream. They can be long enough so that the organisms can divide and respond. And that aspect, I think Severine et al. Managed to capture. And the other interesting aspect, which once she gets her router rebooted, hopefully we'll hear about, is depending on how violent these submerged vortex rings are, you can either get sort of fluid-like behavior, somewhat like the droplets you'd see on a spider web, the dew droplets. We seem to see that with various genotypes in each droplet. And then if it's more violent, the colony that's sort of floating on the surface can't respond as if it were a liquid of dewdrops, say, but it actually fractures. Again, a little bit like continental drift. And the continents break apart. She may get to this. Each continent itself, it seems, has a submerged vortex ring underneath it in this little dish.
Wow.
So again, that's something we definitely need to understand better.
Right. Someone who's attending is asking, it looks like Xiaomin is asking, what is in the media to increase density and viscosity?
Andrew, do you want to take that?
Yeah, it's a it's a form of modified cellulose, and you can. It's sort of like it's effectively like putting cornstarch in. It's just a way of
Oh,
super thick.
I made it.
oh, hooray!
I'm so sorry. This is a terrible timing. I have no idea what happened.
I was just saying to folks that is the ultimate nightmare. You know, it's akin to your train or your bus or your flight being late, but in virtual world.
It's funny, I had a dream about it.
Really? Oh no.
Yes.
Well, I'm sorry then you've had to experience it twice. But we were actually just having some great conversation here about some of the background and the questions that set up the work that you did and the project. And so we were just talking about what was in the media to make it have greater viscosity, but you also feel free to jump in and with your presentation, and then we can return to some Q&A afterwards.
Sure.
That sounds great to me. And Severine, you should know that we began with your lovely music video.
Oh, great.
I want to show that at the end as well, just so that people can close the loop. But you decide, and I think it would be great if we could turn the stage over to you.
Okay, great. Yeah, I can start sharing my screen.
Sure.
Late joining. I'm really happy I was able to make it. I'm still under the the adrenaline of fixing my connection. So hang on with me here. Okay. So I first would like to of course thank MSI and Anna for reaching out to give us the opportunity to talk about our research in this seminar series. I've watched some of them, some of the previous ones. They're really interesting, and I think our work seems to be fitting very well in that spirit. And I also would like to thank you, Andrew and David, for joining today to enrich the conversation. So the story, as you probably have seen a bit through that video we made for the APS contest, is about yeast colonies growing on liquid surfaces. But I'd like to give you a bit more feedback about this story and like how it started and what were the motivations behind. And so one central questions that we can have is one related to evolution, the questions of how did we get here and why are the organisms we see today on Earth are the way they are, and how did they evolve? And this is of course a very deep and vast question that everybody's interested in, not only biologists, but physicists, mathematicians, chemists. And so if we have the general approach with, for example, looking at the tree of life, on the outer boundary, we have the species that we see today. And then as we go toward the center of this phylogenetic tree, we see that those branches, those different species are connected to a common ancestor. And as we go back in time, they're connected to less and less ancestor until in the center, we reach this universal common ancestor. And we cannot replay this film back. This experiment was run only once on our planet. And so we don't have any way to, unless we can travel in time, to to see it and reconstruct it by observation, but instead we use genetic correlation, we use fossils and multiple ways of what we have today in hand to rebuild this tree of life. And I would like to point that this is actually a very similar endeavor in a very different field of astrophysics and cosmology, where we are trying to rebuild the story of the universe. We're trying to understand the origin of the things we see today. And we also don't have a way to replay the tape and understand how the structures and the galaxies we see today have been taken the form they have. However, astrophysicists have a way to cheat. They have light. And so because light propagates at a given speed, then the further we look in space and the further in the past we can see. And so this is a way to time traveling, and this is a way to basically rebuild the spatial temporal evolution of the universe. And what I would like to show you next is that we can actually do something similar, rebuild a spatial temporal map of evolution in a laboratory experiment. So this is one way of course, approaching these ideas of evolution and competition, but this is the one.
I'm so sorry to interrupt you, but there's a little color panel up in the upper right, and I don't want it to obscure any of your future figures.
Sure. I think, let me see. This is one of the classic one. Of course, it is appearing someplace I can't see it. Let's try.
It's pretty much out of the way, but I just wanted to let you know.
Okay. How about now?
It's still there. For me, it's in the upper right, but maybe let's see if it reappears.
I can try to, right, let me try again. All right, how about now?
Perfect, looks great.
Yes. One way of going over this type of phenomena in the lab is to look at smaller organisms that can divide faster. And so here I'm giving two examples of this type of organism. We have E. coli, that is a bacteria, and we also have yeast, that is a eukaryote. And so in this case, those organisms can divide fairly quickly in the timescale of a lab experiment, so around an hour, and so lead to multiple generations. So we can basically on a Petri dish grow several thousands of generations of these organisms in timescales that are accessible to a typical couple days experiment. And so this organism present this advantage, and although we cannot redo the crazy complexity of evolution among all the species, we can maybe try to understand the very minimal ideas and mechanisms that can be present in the more complex real size, real life system. And the way physicists sort of see this type of experiment is we have basically particles that can divide, that's what lead to growth. And they can also disappear because of a limited amount of available nutrients. And then we have some diffusion that could represent the fact that the cells are not exactly at a given position over time, but they can jiggle a bit around because of the neighbor pushing on them or because of some thermal fluctuations affecting them at this scale. And the next thing we can do is to color these organisms to follow lineages. So by that, I mean that this is something very common in biology. We can basically genetically modify these organisms, and this way they can produce a fluorescent protein we can then track under the microscope. And in this situation, when we have a given protein, a given fluorescent marker, then the progeny of the cell will also carry the same type of fluorescence. So this way we can follow basically the progeny of the green particle and the orange particles in this example. And if we do an experiment where we have several of these, and so let's say we start with the exact same type of bacteria, and we choose 3 different colors, 3 different genetic markers, and we come and inoculate 1 droplet containing a third of each of these bacteria on in the center of this petri dish. And we look over time how the colony is growing, and under these different fluorescent lighting, we can then identify those different type of bacteria. And so here you can see the time scale is in hours. So within a couple days, we can actually even within the first 12 hours, we can see a segregation happening. We can see the formation of these genetic domains that are only one type of bacteria. So if we zoom in, we actually see that there isn't any averaging in this region. So maybe closer to the boundary of where the droplet was deposited, we can still see some multiple colors, but very clearly after some time, these bacteria seem to be separating. And so that tells us something very interesting. That tells us that competition is very important in this geometry. And a way we can explain it is the fact that growth only happens at the periphery of the colony. And so that means that available nutrients are only accessible to the cells that are at the edge of the colony. So what's behind doesn't really affect what's happening next, in the sense that now we have a very small number of cells that are going to compete and decide of the fate of the next generation. So if we look at these schemes, we can see that after some time, this will naturally lead to a sort of coarse graining, a fact that fluctuations, the fact that the green or the orange particle made it first, is going to affect basically the rest of the growth here. And so this is typically due to the fact that it's a small population that controls the growth. And so in that case, fluctuations are going to have a strong impact on the rest of the growth. And so this has been observed some time ago and is actually quite universal. We can show that it happens in bacteria, E. coli, but it also happens in yeast. And we can understand this because it's not something that depends on the specific biology of the cell, but is more something geometric and mathematical. So we can explain it, and it can apply to different species as long as they are in the same configuration. But this is not the whole story. So I showed you one way of sort of replaying the tape of evolution. So if we get back to this type of experiment, this is what I meant by looking at the past in space. Is basically this type of pattern allows us to see how things have evolved over time. So if we look at the periphery of this colony, we see segregated regions. But now we have an experiment where we know how it started. So by looking toward the center of the colony, we're basically going back in time. And so there are lots of things that we can do with these experiments and try to see if we can extract even more physical mechanisms, biological mechanisms to understand globally how this type of system can generate segregation or those type of patterns, and can we see them in nature and understand something from this type of model systems. But if we think about nature, the environment is never homogeneous or still, and this type of competition actually happens in quite complex and time-changing environment. And so if we want to have some image that's more realistic, we also need to take into account all these possibilities. And so here I'm giving a famous example of the plankton. Plankton grows on the ocean surface current, and in that situation, the growth and the competition between phytoplankton is going to be coupled with hydrodynamic flows at the surface of the ocean. And so this is going to completely change the way these species, basically these organisms, are evolving. Just to illustrate with some more examples here, we can, so in this work on the left, we can extract the shape of these ocean surface flows. So this is a satellite measurement. The grayscale basically show some chaotic mixing structures that are naturally present in the turbulent flow on the surface of the ocean. And by releasing some tracer particles, so some markers here that are going to follow these flows, this is numerical simulation in this case. By coupling it with this ocean surface flow, we can show that there is a segregation happening due to the shape of these hydrodynamic flows. So this mixing is going to affect how different species, for example, or different phytoplankton bloom are going to interact with one another. I'm giving another example of the Black Death propagation in medieval Europe. You can see that this is also a growing front, exactly like the bacteria colony. But in this case, it's the spreading of Black Death, and it is also going to be strongly affected by the environment, by the topography, by how people are mixing. And of course, black in the medieval time, we didn't have any planes, we didn't have any flights, so things were quite simple in that case, and we could see a propagating front like this. We can think of forest fire. It's also propagating front. That's going to depend on the tree distribution and the topography. And here I'm giving also another example of the chemical reaction that creates the ozone hole in the atmosphere. And so this reaction is going to be coupled with atmospheric flows. In this example, they're showing these vortex type polar atmospheric flows that seasonally appear in the pole. And so that's also going to completely affect how this chemical reaction is going to propagate and thin the ozone. Okay, so if we get back to the idea of how this type of transport phenomena, more specifically, so if we focus on flow examples, how is this going to affect the formation, let's say, of these genetic structures I just shows that appears in those Petri dish experiment? And more particularly, what are the fundamental mechanisms? Can we understand how flow is going to affect those structures, and are there like specific type of couplings that are going to generate a different scenario of evolution? And in hydrodynamic, we can basically cut down complex flows like turbulent flows, chaotic flows, into simple structures that basically are the building blocks of a flow, of a more complex flow. And two examples here are what are called hyperbolic points. So this is a point where if we have a blob of material coming into that hyperbolic place, then it's going to be stretched away. And another typical structure is an elliptic structure where if we have a blob that gets into that, let's say a vortex, it is going to undergo shear, and so it's going to also spread, but with a certain type of shear, or become encapsulated in that elliptic regions and be isolated from the rest of the flow. And there are also evidence on the theory side that the shape of the front, so if we start deforming those colonies, is going to have a strong impact on how competition and selection is happening. So in this example, without explaining the mechanism in detail, if we mark every cell with one color, so if we had a rainbow type of lineage tracking in the experiments, then this is what we would see. So we start at the bottom with all the colors, so one cell, one color, and then we let the colony grow. And depending on the type of growth we allow in this model, so one on the left that leads to a more rough front formation, and the one on the right that leads instead to a flat front propagation, we see that the shape of the front is going to change the genetic selection. And you see that on the left, we have much less colors, so larger domains of one common ancestor. And the one on the right instead has more diversity. So there's clearly an indication that this type of coupling with transport deformation is going to have an effect on evolution. And so I come to the project. I had the chance to work with this wonderful team. And so I think you already met David and Andrew while I was trying to desperately connect. And also Brian Weinstein, that was a graduate student with who I worked and was also very active on the project. We basically did everything together, and he also did the numerical simulations that I'm going to show in this project. So this is the ideal experiment we wanted to do. This is how we started this project. We wanted to basically have the typical Petri dish experiment with a colony growing on top of a gel. But instead of the substrate to be solid, like in the case of agar gel, we wanted to have a liquid media. Because with the idea of basically putting some syringe pump flows, mixing the media so we can slowly deform the colony while these genetic structures are appearing. And so we can study how those hydrodynamic structures can affect the selection. But we had to begin at the basic point, which is first we have to be able to grow a colony on top of a liquid media. And that was actually a big challenge. It's not easy at all because the first problem are thermal fluctuations. That why nobody has done this before in lab. It's because we are asking something that's a little difficult in terms of fluid dynamics. We're asking to have a fluid that is not turbulent, that doesn't have any convection for very long and extended period of times, because we want to see those genetic structures develop. And if we look at typical colony front growth velocities, they are the order of micrometer per minute. So this means that the flows that are present shouldn't be faster than that. Otherwise, we're going to mix everything and we won't be able to do the same experiment. So I thought, okay, maybe a way to avoid this is to increase the viscosity. By increasing the viscosity, we increase the dissipation. And perhaps we can this way kill any type of undesired thermal convection that would otherwise, in this type of context, so at the scale of a Petri dish, mix everything within 10 minutes. And so I added polymers to the growth media and reached a viscosity that was 1,000 times the viscosity of water. And so this is what we saw. This is a droplet of inoculant that contains E. coli. And the time scale is about 12 hours. This is what we saw, a disaster, which is basically a gigantic convection cell of E. coli bacteria growing like a tsunami over the entire Petri dish. So no genetic domain formation, everything is mixed. So even at 1,000 times the viscosity of water, we couldn't have a still environment enough to see this colony develop. So I went ahead and increased it more. And once we reach 100,000 times the viscosity of water, which is really viscous, this is comparable with honey, just to give you an idea of what it would look like in lab. I'm showing here a colony of yeast. Then things seem to be growing normally. So we did it. We managed to have a liquid substrate on top of which the cells can grow, and the absence of hydrodynamic flow externally controlled, then we don't have any perturbation to the colony. So at this point, we were happy. And this is what it looks like to drop an inoculant of E. coli on the surface of this substrate. And if I zoom in, you basically see the individual yeast cells that are relaxing and staying on the interface. So this is good. They're not sinking in the media either. The surface tension is basically holding them. And they form this little raft due to those surface tension interactions. And so everything seems to be normal. And if we wait now long enough and look at the growth of the colony, then this is what we see. So we see the colony expanding. We also are tracking here with two fluorescent marker, the lineages. So we have, in this case, two different types of yeast that have the same fitness, so they grow at the same speed. But you can already see that the domains doesn't appear immediately, like on the gel experiment I showed, and they also look a little different. So if we do some more quantitative measurements here to look at just the basics of what's different between growing a colony on top of a gel substrate versus on this new viscous substrate. So on the right, I'm showing what we would see in blue circles on a hard agar plate. This is the radius over time. And the green squares correspond to what we see in this medium. So at the very beginning, we have this exponential growth that's leading to a much faster expansion of the colony than its counterpart on the agar gel. And then after some time, then we see that the velocity, so if you look at the lower scheme here, the lower plot, you see that the velocity reach a constant and then start decreasing. And exponential growth is interesting because this means that what I said earlier about the cells only at the frontier deciding of what's happening is no longer true, because if we have an exponential growth, this means that the entire colony is growing everywhere. So if I zoom in and show you what it looks like in these early stages, it really doesn't look like it's only the front that's growing, but it's instead the whole colony is expanding for 12 hours in this case, in this movie. And it really looks like a sort of expanding balloon in this movie. And so once we slow down, you see at the end of this movie, the growth seemed to slow down, probably to some nutrient limited growth, the fact of after some time, the nutrients available under the colony are going to decay, then we see that the colony slows down. and the velocity almost reached the same velocity as on an agar plate. And very briefly, this type of two-stage growth is also observed in other organisms. It has been seen in lichen, where it is shape dependent and depend on the influx of nutrients. So at the beginning, on a spherical colony, the influx of nutrient is homogeneous, so the whole colony grows. And as the colony flattens, as it at later stages, it forms a more pancake-shaped growth, then the nutrients are going to be mainly, the flux of nutrients are going to be maximized at the edges, and the colony grows mainly on the edges. So we think something similar to this happened in our substrate. But to further this study, I added some fluorescent beads in the substrate just to check that if there's any flow happening, or like if the motion of the substrate plays any role in the way that the colony is expanding so fast at the beginning. And this is what we saw. So this is the fluorescent particles in the substrate that are basically moving away from the colony, radially out. So this is a long shot of the type of motion we recorded. And if I do some particle image velocimetry to measure the velocity, the flow velocity field, this is the type of field we see. And what I would like to say about this field is it is much faster than the colony front speed. So it's not just the colony pushing on the substrate, but instead it seems to be something else that is going much faster than the colony growth rate. And also, if we look at where the peak of this velocity field is, the maximum amplitude. So on the right, I'm showing a radial profile of this velocity field. Then we can see that, this is actually further away from the colony edge. So something is making that convection. And that was a little disappointing because we thought we got rid of all the convection, and here the convection is back. However, it doesn't show up. It doesn't show up if we don't have any colony growth. So at least we thought, okay, this is something that is generated by the colony itself and not through uncontrolled lab conditions. And we have a lot of ideas of what the origin could be. And so we had to go through a bunch of tests in order to understand the origin of this flow. And so we did basically all the orientations imaginable, because we were wondering if that would reverse the convection direction. I attached the colony to a gel layer and then submersed it, so it was a sort of upside down configuration.
Hi, Severine. I don't know if it's just me, but I can't hear you speaking anymore. It looks like your mic might have gone out. Oh no, I think we've lost her. I wonder if it's an internet issue. Okay, we'll give it just another minute here to hope that she can sign back on with us. And if not, then I'm definitely going to ask for, maybe we can get a recording of the rest of the talk, because I don't want to leave us all hanging this way. So let's just pause for a moment and see if we can get her back here for some concluding remarks. I'll be back. Okay, I think that given the time, I'm going to go ahead and say that we should wrap up and sign off. And I'm so sorry for the technical troubles. Unfortunately, this is just inherent to all trying to do all the things that we want to do and interact in all the ways we want to interact in a virtual space. And so I'm glad to have at least heard this portion. And like I say, I will probably try to meet with Severine again and hear the rest of the talk, and we'll record it and make it available to all of you who maybe we'll be able to come up with some Q&A and more of a chat space or an internet space at that stage. But thank you all so much for attending and for bearing with us. And also, relatedly, if you would like to email me any questions to pass along to Severine to be answered, or any curiosities you have, then I see a couple coming in here, and I'll make a note of those and be happy to pass them along.
Very good.
Is there any?
Thank you all for coming on Severine's behalf, and I'm very sorry that this internet issue intervened. But maybe you could post the paper, or if there's another talk, we could have a pointer to that.
Absolutely, yeah, I think so too. That sounds good. Thank you so much for joining. And also, before everyone leaves, I'll just put in a plug for the fact that MSI is really excited to do a lot of collaborative and interdisciplinary seminars this year. Our first one in January was with Barn, and this one is with folks who have worked in physics and MCB, and we're looking to carry that out in the future. So we're contacting a lot of people, but also if you have something in your department or program or school that would make sense as a co-hosted or interdisciplinary talk with MSI, please reach out to us. We would love to do it and talk with you. All right, with that, I'm going to sign off. Thank you all so much. And thank you again, David, and we'll talk to you soon.
Bye now.